Greetings to everybody. First, I would like to thank to the organizers who made possible this marvelous meeting and for having invited me to here. In my present speech, I will try to tackle the somewhat long or 160 years long mystery of the origin of life. That perhaps is such a mystery only because our contemporary biology is so molecularly reduced. It looks on life through the very narrow molecular glasses. And I will try to challenge this outlook that it was very difficult, if not almost impossible, to get the organized life once in the history, and that perhaps there is no life anywhere else in the universe. I will challenge, as I told, this idea, and I will try to push another idea that perhaps the life is emerging all over, in our vicinity, on the Earth, all the time. So, let's see for a short time the origin of life problem. Until 1859, the origin of life was seen as no problem at all. The organisms were seen to either propagate or at least the simplest one to form spontaneously from the inanimate matter. Then there came Pasteur's famous experiment. I think you know it because I think that we learned about it. He subjected the cooked soup into the glass with a wavy tube so that no bacteria could enter it. It was possible, but he was very lucky, so no bacteria entered it. And after 3 days of monitoring this soup, it was evident that bacteria did not develop there, and so this famous proverb, omne vivo ex vivo, started, so that all life from life. Now, because we know that at a certain moment in the history of the Earth, there was no life, it was impossible. And we know that today there is life. And if Pasteur proved that life can originate only from the already existing living beings, now how is it possible that somewhere in the history of the Earth life originated? Many propositions originated or started at that time. So after Pasteur's experiments, certain of them saw the origin of life only as a very improbable chemical accident. The other saw this as some deed of force majeure. And there, of course, were also theories that tried to dismantle this mystery by referring to self-organizing capacities of matter, based, of course, on certain laws. So this was a, let's say, true scientific approach. Now, among the proposed solutions, we had some, let's say, branches after Pasteur, mostly in 20th century, like cellularists, Oparin, Fox, I will come to them later. Then chemists. Of course, this chemical approach was very huge. I mean, it covered and is still covering 99% of these attempts to disentangle the mystery of the origin of life. There was, I think, very well-known experiment from Miller when he subjected the mixture of supposed primordial gases to electrical discharge for 2 weeks and to water vapor. Then after 2 weeks, he got many amino acids. So this is very important precursors of proteins. And this was a lucky day for this origin of life science. But it is interesting that in 1999, he published an article in which he was very pessimistic as to this chemical approach because science did not yet achieve to get L and D or only L amino acids or D sugars, so optically active. And there were many other problems. I will not go into them today. Then we have also theoreticians like Manfred Hagen and today already mentioned Stuart Kauffman, who proposed certain theories how life could start it, just on theoretical and mathematical grounds. And we have a very interesting proposition by Paul Davies in 2014 about the self-organization capacities of information. This is a theory that life is not based on primarily on molecules, but on information over matter. It's interesting, but I will not go into this today. Then we have also some other theoretical approaches or more practical approaches that are written here. Some articles or experiments by Reich, De Meo, by me, by Professor Pollack. Now, let's see. If we want to understand the origin of life, we must first know what life is. And here, perhaps for some of you, first disappointment, there is not yet any universal definition of life. So no scientific consensus. And so it is difficult to say on this ground that we don't have any sharp definition if prions, which are highly infectious agents causing mad cow disease, I think that you know them, they are 13 nanometers only long entities, proteins actually, are living beings because they reproduce themselves and have some similarities in this, and they are highly infectious like parasites. Then what about viruses? Are they alive, or do they only share the life of their hosts? And I think not very well known, but studied exotic proposed pre-bacterial forms like nanopes. These are tiny filamental structures found in some rocks in Australia. Some interesting aggregates, also similar to certain remnants from Mars in Allen Hills. Then nanobacteria, which are regarded as propagating calcified nanoparticles and which are, let's say, hosts or whatever, even in our bodies, in blood. I will show them a little later. And it is interesting that certain well-established scientists from Taiwan and from America connected these nanobacteria to Wilhelm Reich's bions and even named them bions, which I found really surprising in the positive sense of this word. Now, what about bions, already mentioned, that were investigated and named by Wilhelm Reich and further research by his followers, or Béchamp's microzymas, or Bahadur's Jivanus. I will come to these entities later. So you can see the bionts here and here. There are certain aggregates. They are cellular-like structures, smaller sometimes than normal cells or even bigger. And what is interesting is that Dr. James DeMeo, who already lectured in some of these water conferences, reports about moving creatures similar to amoebas or paramecia acquired from totally sterilized but not anorganic stuff. So it is published here, and I have also personal confirmation from him that this is true, you know, even if it sounds very weird. So now what about microzymas, already mentioned, that were found in 19th century by French scientist Antoine Béchamp, contemporary from the already mentioned Louis Pasteur. I hope you will see this interesting particle. This is erythrocyte, this is, I think, a leukocyte, and you see this is there are very living and fast-moving entities. Okay, one would say perhaps this is Brownian movement, but what is Brownian movement is still a debate. And I will not go into, just read the book Fourth Phase of Matter about Brownian movement, I will see. So now let's try to come to some sort of definition of life or characterization. We can see life as a highly organized and dynamic process involving information, energy, and matter. So not only molecules, which would be covered by matter, but information, energy, matter, IEM, capable of autonomous continuation and potentially limitless evolution by means of differential reproduction of its basic units. These are organisms. Now, if you look on this ribbon, which represents time, and if you look on life from more standard biology, then we should say and this is a start of the Earth, we would have something that was not life or suborganized, IEM, these are information, energy, matter. Then some transitional systems, some transitional chemical system, at least this would be seen by contemporary biology. Then somewhere, not sharply demarcated origin of life and life. So now, if you look a little more closely to these transitional systems, if we are still in this more biochemical outlook of life, it would mean the development of a complex and highly efficient molecular systems resulting in the genome and genetic code out of purely chemical, supposedly autocatalytic systems. And I would like to challenge this view by another, which is not biochemical transition, but physico-chemical transition, based on specially organized states of water, water solutions, and various agglomerations in water. Now, let's see these three basic stages of the origin of life, covering this transitional, transitional IEM. This would be water cluster stage. I think that we heard a lot of this organized water now, and of course previous conferences. Then the second would be cellular or vesicle stage, and the third biochemical stage, which would mean that we went from certain organized vesicles to modern biochemistry and genetics. If you come to this first, this water stage, I think that it is not difficult to imagine that during the prebiotic Earth, there are many hydrophilic surfaces at hand, various clays, colloidal systems, agglomeration of organic stuff, and so on. So this stage did not need any special and long-lasting evolution. Therefore, it is logical it was the first act in the origin of life drama. According to the contemporary new water science, you would expect easy water system around surfaces or around oblong as well as spherical aggregates, and then various coherent domains in ferroelectric modes or around ions, and this basic coherent, already mentioned 100 nanometers quasi-spherical coherent domains with no solutes. So many possibilities of ordered water that this stage was, I would suppose, very quick. It started with water, with liquid phase, when the Earth cooled enough. Let's see some of these lifelike features of water exposed to hydrophilic surfaces that can be called water hydrophilic systems. We know that all life needs free energy that is capable of work. And from the research of easy water, we know that this interfacial water system can draw the environmental radiant energy and transform it into electrical potential. And this energy can later be transformed into mechanical or chemical energy, even much before photosynthesis or organized metabolism. This is very important. So we can have certain very important features of life before this very, very complex organized biochemistry. Then the second thing is that all life needs orderliness. And exclusion zone research demonstrated a complex organization of colloidal systems in the vicinity of hydrophilic surfaces. Look here is a wedge of many times mentioned Nephion, and how a colloidal system behaves in a very orderly fashion, even 3 millimeters in 3 millimeter distance from the wedge of Nephion. And this is from our research. It was presented in previous year. These are highly ordered structures of water evaporated and put under, these drops are put then under dark microscopy. And you can see that these are drops from spring water and a drop from the same water that was treated by a healer on some distance. And you can see that the pattern of these drops is quite different from the ones, but the water again was the same. So again, we can gain a quite different order in water demonstrated by this droplet evaporation method. The second stage, as I call it, was vesicle stage. And it demonstrated electrical activity over the membrane, agglomeration of particles, so making aggregates, reproduction, mobility, and complex long-range orders throughout the cell. Now look more closely on this. Electrical activity over the membrane was found in so-called proteinoid microspheres These protein microspheres were studied by Professor Sidney Fox, I think that even in 50s or 60s, and until 80s of the previous century. And they are similar to, I think, more known coacervates, pairing coacervates. These are aggregates of protein-like molecules. And what was striking was the finding of Japanese group. It was published by Ishima in 1981 in the Biosystems. And these are quasi-action potential from alkaline acidic microspheres, and the voltage was around 50 millivolts. Again, with no specialized biochemistry and no long evolution. This otherwise is a very complex and specific physiological response of excitability of modern cells. Then agglomeration, we already heard from like attracts like mechanism, and this we can see from the study of Oparin's coacervates. Oparin actually proposed, this was a great Russian scientist, Alexander Oparin, he proposed the first true hypothesis how life could emerge in a convincing way. And coacervates are colloidal formations resembling cells and showing many similarities to present-day living cells like primitive metabolism, growth, budding, division, aggregation, and so on. So here are certain specimens of these aggregates. And we can come to Reich's bionts, already mentioned. I don't know into detail here. We can just see that if we put dead moss or straw into water and it is autoclaved or sterilized, we see that it decomposes into smaller particles, smaller from the cells. It was called Bionose disintegration by Reich. But after several weeks, this autoclaved grass water infusion develops numerous highly organized forms which appear to emerge from the background sea of Bionose aggregations. You can see these formations here. And later it is said that, as I already said, even protozoan-like moving structures may develop, which is striking. And in our laboratory, we are just beginning to tackle this research. And then we can come to Bahadur's Jivanus. These are a grain these show agglomeration, growth, and reproduction. And these Jivanus are formations, aggregates that are formed from mostly anorganic material that is exposed to sunlight. I will come to them a little later. Then I think that from many presentations of easy water, you know that about the movement of microspheres because the surrounding easy water, and they are moving towards the source of infrared or visible light. And it is, I think, already very well known. The flow of bulk water that is surrounded by easy water and oriented flow of water from above downwards around Nafion beads, for instance, is also known. So if you have organized water like easy, you can have also directed flows and movements, which are, of course, known also from organisms. And again, this is a presentation from our laboratory of this the so-called bionts. I don't know if this is seen here, certain movements. They are not very striking, but you can see these formations or these ones. So when you look through the microscope, and this was taken just from autoclaved moss and straw, and it developed very quickly. So let's come to the last biochemical stage. So we know that various organized vesicles like conservates, macrosphere, and so on demonstrate a rich inner microstructuring or differentiation, and mostly they have interfacial, so highly ordered water. And we may expect diversified, abundant, and numerous coherent domains in this water. And also from other research, we know that coherent electromagnetic oscillations can couple to macromolecules and to many molecular interactions and chemical reactions. Two giants of science of coherency, one is Emilio Del Giudice, the other is Herbert Fröhlich, wrote about this connection between coherent domains and coherent states and macromolecules and molecular interactions. Actually, Herbert Fröhlich, in his cancer theory, proposed that coherent regime controls even such biochemically complex processes as a cellular division. So this is very important and interesting if we take into account that we can have very rich complexes of different coherent water ensembles in this conservates microsphere and so on. Now, if you put this together, so Fox microspheres that show very complex biochemistry and even some forms of protein synthesis around RNAs, then Jivanus that were found then composed even of amino acids, sugars, RNA and DNA monomers, and ATPase and peroxidase activity and RNA-like molecules stemming from very simple organic and inorganic molecules and minerals after exposure to sunlight. And Reich's and other experiments involving bions and resulting in protozoan-like structures, we can infer two conclusions. First, that it seems that a complex biochemistry does not need billions of years of slow evolution via a selective process to develop enzyme-like molecules. And second, that instead of this, a reinforcing process between a highly complex, ordered intravesical milieu and molecules, different molecules and their aggregates, can bring an abundant biochemistry very, very quickly. Now, the conclusion. So, the question, how life emerged? Taking into account all experiments known up to date, we can consider it a wrong question. It should be reformulated into, is life constantly emerging on the Earth? Of course, it depends on the definition of life. Now, if you look again on these transitional information, energy, matter systems, first, there was a water cluster stage. We know that it is permanently occurring on the Earth. Second, cellular vesicle stage, again, permanently occurring. Biochemical stage may be developed much faster than thought hitherto, as I told before. So if you identify life with developed vesicular water structure that have capacity to develop further into more sophisticated biochemistry, life is constantly appearing, though it represents a different form of life as understood by contemporary biology. It has no genetic code and is highly variable, this means pleomorphic and chemically versatile. But regarding ordinary organisms and biology, the essential question deserving concerted effort of different laboratories would read as follows. Is it possible, and does it happen, that organized vesicles can shift into life forms that would be, one, unquestionably classified as organisms, and second, might be even identified as some of the known species? And even if the answer proves negative, the organized, ordered, water-infused, supramolecular aggregates would deserve their own thorough research branch. All the discussed lifelike structures strongly indicate that water and its numerous short as well as long-range interactions with organic and inorganic stuff are more important for life than DNA. The latter may be regarded as the last step in the biochemical phase where specialized enzymes and their highly efficient complexes, including the genetic code, emerged. Thank you for your attention.
All right, we're running about 15 minutes behind. Let's take a few questions, and then we have a brief announcement before we break.
Are you familiar with the work of Andrew Cross and his abiogenesis of Acari insects? He used a methodology that would seem to indicate he was generating easy water using a strip of flannel that was used as a wick to suck water up, and he was attempting to synthesize artificial crystal growth using weak electrical currents. So it seems to absolutely support your research, and it was later replicated by Michael Faraday. I suggest you look into it. His name is Andrew Cross. He was in the 1800s.
Okay, thanks.
I would like to know your opinion. Thank you, first of all, for your talk. I would like to know your opinion about this, the highly repeated and highly successful structures that you find in biology. If life does not come from an evolutionary path, then how are these evolutionary conserved structures, the so-called evolutionary conserved structures, evolve and come to exist from bacteria to us?
I didn't understand. Which structures?
Any kind of highly conserved structures in our organisms from a biochemical point of view, like a potassium channel or any other macromolecular structure.
So what's your question?
If you said in your talk that life does not need an evolutionary path to come, like from millions of years, so how do they come to exist in all of us in the same way?
Well, I would say that it depends on the definition of life, as I told already. So, okay, I'll go ahead. Of course, there was the path of billions of years of the evolution of life, and I think that the genetic research here is undeniable. But I'm speaking about certain forms of life that are inchoate, not yet really formed, but organized, and that have all basic features of life, So we can have certain, let's say, primitive or whatever life. Life there is not formed in this specialized, highly complex biochemical sense, that we know life today. So there was a long evolution, biochemical evolution, but we can have life even on a much lower level of biochemistry, of specialization of molecules, and this life can be emerging even now, around us on this level. But then there comes very slow evolution of biochemistry with the genetic code. Genetic code is amazingly complex, immensely complex. So there is no problem. The problem is the definition of life, I think. If we identify life with DNA and other macromolecules, or if we can see life also in organized matter without these molecules. Okay.
Do you think that the amino acids and the proteins and even the nucleic acids arose later, and that their form is such that they could fit into the matrix of water structure? Do you think that was a determining factor in the structure of these macromolecules?
Yes. I think that there should be some correspondence between macromolecules and certain and some ordered water. I mean, certain, let's say, domains or easy water and so on. I think that yesterday Elia spoke about this correspondence between beta sheets and some ordered water that he found. So this is an interesting example of this, and perhaps that was also found something like helical structured water or so, you know, in certain experiments. So yes, I think that the ordered water in certain ensembles or coherent clusters can be similar to certain macromolecules, so that we can have something like resonance. Okay.